ReNeuron in Wales. He's here to talk to us about the development of human neural stem cells for chronic stroke disability. Thank you.
Great, thanks very much, and make sure I've got the right thing. It's working. Yeah, and thanks very much, and thanks to the organizer for inviting me, and especially to Frank for inviting me to come today. So, as indicated, we're a stem cell therapy company. We're based in Pencoed in Wales, near Bridgend, as of about 3 years ago. In terms of we're a clinical stage company, so our pipeline is here. We have our lead program is which I'm going to go into some detail here, is in stroke disability, and we're currently moving towards a phase 2b pivotal trial. So this will be a randomized control trial, and I'll talk to you about the trial design. We also have done a phase 1 trial in the UK in critical limb ischemia. Now, CTX is our main lead asset. It's a neural stem cell line, CTX, because it's derived from fetal cortex. And from this platform, we also have now developed quite a lot of work with exosomes derived from this producer cell. And we're following in particular in various cancers, and we're in the process of going through quite a lot of preclinical and in vivo and in vitro data to look for a potential cancer indication that we wish to target sometime by the end of next year. And more about that later. We also have a second asset. These are our human retinal progenitor cells, originally in license from Michael Young at Scape and Zai Research Institute. And we've moved through all preclinical through into completing a phase 1 clinical trial at Mass Eye and Ear Institute with Dr. Eric Pierce and Jason Commander as the surgeon. And we're also looking to start on the back of the safety data coming through from the retinitis pigmentosa trial to also do a trial in another inherited condition, cone rod dystrophy, which is kind of the reverse. Retinitis pigmentosa is where you lose rods and eventually cones. Cone rod dystrophy is the reverse. You lose cones first, followed by rods. So I'm going to focus on the CTX cell product and where we've got to clinically with it. So this is there's a lot about manufacturing on today. So this is really our sort of roadmap in visual terms to production of CTX cells. And it really began 20 years ago when we first started isolating human neural stem cells from fetal brain. Through that, we then moved to generate a technology that would allow us to expand these cells in a means that we think could be clinically useful. And I'll explain that in a second. Once we got these, what we wanted were clonal cell lines. So we got those clones and we tested them. We demonstrated in vitro phenotype functionality and also in vivo, looking for survival and differentiation in vivo. We then had to scale up to look for patient indications, and that involved quite a lot of process development and a lot of analytics. And from there, we're looking to manufacture it at scale. Most of what we're doing now is in TFLOS, so therefore automation is one prospect. But we're also looking now at, we have we're participating in a grant that we're leading with the Cell and Gene Therapy Catapult and Loughborough University to look at all of the different cell stack systems and bioreactors applications that we can for CTX cells. We've also developed a formulation, and I'll go through that in detail, and we are actually working at the moment with a frozen formulation. And this is a formulation that doesn't have DMSO in, so the cells do not need to be washed prior to use, they simply need to be thawed. And that's basically, you have to then have a full quality system over all of these processes to be able to go into the clinic. And this is one of our patients who, we often get patients appearing on breakfast TV at the BBC. So this is the key to the technology is conditional immortalization of these cells. And what we use is a fusion gene, which is c-Myc, fused with a modified version of the estrogen receptor that responds only to one ligand, and that's 4-hydroxytamoxifen, which is a minor metabolite of the drug tamoxifen. So we introduced this fusion gene into dividing cells using a retroviral, Maloney retrovirus system. So that means that we can take up into a random site, single site in the genome for this fusion gene, and that fusion gene then makes a fusion product that will respond to 4-hydroxytamoxifen. So we can use 4-hydroxytamoxifen to specifically expand those cells that have the gene and making the product. Then we can take those cells off. We can start the process of cell banking by making seed banks in-house. So this system then is an effective system. Basically how it works is if these cells constitutively express the c-MYC-ER gene, now the c-MYC-ER gene is present, obviously, so inside the cytosol you've got the proteins. These proteins can only enter the nucleus if they're dimerized, and the only ligand that can dimerize c-MYC-ER is 4-hydroxytamoxifen, this particular variant. So it comes in, it activates the cell cycle, and when you remove 4-hydroxytamoxifen, then you cease to have any c-MYC-ER active in the nucleus. So it's present in the cytosol. And furthermore, when the cells are implanted, you actually get epigenetic silencing of this gene because it's a Maloney insert and it's in a particular location. It allows for gene silencing, and that's done by methylation of the promoter element. So I'm going back here to 2003 when we generated our first set of clones. So we ran a campaign. We generated over 100 cell lines from cortex and striatal regions of the brain. We ran a screening then to, if you like, a kind of drug discovery or a cell discovery program, where we ran in vitro assays for things like freeze thawing. Are they robust enough to be made into a product? Do they express all the right neural stem cell markers? Can they differentiate into neurons and astrocytes and oligodendrocytes, etc.? All of those things. And then from that, we came down to 16 cell lines that we thought we could carry forward. And then we ran in vivo assays. We put them into various animal models, looking for survival in vivo, some kind of integration, some kind of evidence of differentiation. And from there, we went into animal models looking for any behavioral effects, so looking for functional changes in the animal models. And we came up with 6 lines. And of those, 3 of them are in regular use. CTX is one of them. 2 others are in use in labs all around the world. So manufacturing then has been a huge issue. In fact, most of our staff now, I would say, are involved in manufacturing at some level. And so a long time ago, we made a master cell bank, and we still use that master cell bank of about 150 vials. And from that, from a single vial, we can make working cell banks, okay? And these are hundreds of vials of cells. And from there, we make, from the working cell banks, we make a large drug product. And the reason we can make a large drug product is we do have this frozen formulation. And that means that we can make a lot of product, so we don't have to make it just in time in terms of when it's used. And the product that we're using now is in sealed frozen vials, and it can be shipped anywhere and used. And all of these things require, obviously, a whole raft of assessment showing stability over the manufacturing process. And this is the planned phase 2B clinical supply that we're starting. And again, we're still, as I say, we're still using T flasks. We're using multi-layer T flasks. And we can go in this process through 5 passages in drug product manufacture from a single T3 to, so we're going about 72-fold expansion over a 5-passage period, over a few days. And sort of the evolution of the product in terms of its stability. So in phase 1, I'm going to talk about the phase 1 trial that was done a few years ago, started in 2010. We had then basically a GMPized version of the lab process. And what we came up with is a formulation of cells at 2 to 8 degrees that had a very short shelf life, 9 hours, and in a very standard vial. For phase 2A, we had developed this frozen formulation. So we were able to keep the cells at minus 135 degrees. Our batch size could be bigger. Our shelf life could be much longer, and we progressed it from 1 month to 6, although we were still using a standard vial. For phase 2B, we've massively increased our production size. We retain this same frozen prep, and we're extending the shelf life from 6 months out to 12 months. So that means that you can deliver the cells to the clinical site, and they can use them whenever they need to. We've got a controlled thawing protocol, which brings them up to room temperature. Then they have a 3-hour shelf life at room temperature assessed. So they can be then administered, obviously in this case, through a stereotaxic procedure, which would take a couple of hours to do. So a lot of the work that we're now doing, clinical work, is based on a well-established and published set of data preclinically. And we actually developed, going back to my academic days and my colleagues, we developed a chronic stroke model, which was if you do the middle cerebral artery occlusion, at the time, people were just literally killing the animals hours or days just after that. But if you nurse the animals through the illness of the acute stroke, you can see the animals fully recover their body weight. They become effectively normal in their home cages. But if you challenge them with, with particular tests, you can see that there is a deficit. And the classic one that we use is the bilateral asymmetry test, where you put sticky tape on both paws, and you look to see how long it takes the animal to remove the sticky tape. They find it aversive, they remove it, but they're much slower at removing on the affected side of the body. And you measure the difference score. And so you can see that animals with a vehicle injection here, so this, you do a lesion, you wait a number of weeks, and then you start assessing the animals after the treatment. And you see with the vehicle injection, it's a steady state deficit. They never recover spontaneously. And with different cell doses, you see some level of recovery, but at the highest dose, the 450,000 dose, you see a full recovery to normal levels. And we also showed in another paper that you had to inject these cells very close to where the infarct was. So you had to go into healthy tissue. You couldn't go into a ventricle, for example. You wouldn't get any recovery function. And note how consistent, and it takes a while for the recovery to really kick in, and it's persistent. It goes out to 3 months in these studies. So how does this work? So we've been looking at mechanisms of action. It's quite a difficult thing to do, but we know that we put these cells in, we get survival of maybe 5 to 10% of those survival cells in the long term. The greater the amount of damage, the more cell survival you see, as you might predict. In particular, if you get cortical damage, you get quite good survival. And we inject the cells here in the putamen area, close to the area of damage. There's obviously a strong release, and possibly through exosomes, of growth factors and cytokines, and this modulates the immune system. And you get formation of new neurons, and we publish this data, as well as new blood vessels forming in the ischemic area. So there's a multi-layered approach to healing here. And you might say, well, what about cell replacement? Is that important? So recently we did a study with, so going back a number of years, we used to use the quinolinic acid lesion model. Quinolinic acid into the striatum produces loss of particularly of GABAergic interneurons. And we found that the cells would survive on net neurons in that area. So we did this more systematically in collaboration with Ji-Won Song in Cha University in Seoul. And we looked at injections of our frozen drug product into these QA lesioned animals. And we got nice recovery of function in both a gross motor test, such as the rotor rod, and the staircase test, which is a test of fine motor function. So you get it takes a while to kick in, but it gets better and better. And what we saw post-mortem is that the cells were located precisely where the lesion was. So they they found the lesion site and they implanted themselves there. They made lots of interneuron phenotypes. So we saw GABAergic cells, CTX cells. We saw DARP32 positive cells. We saw glutamic acid decarboxylase positive cells. And we also saw that if you injected plurigold into the anterior striatum, we saw actual connectivity with these implanted cells. So we're getting good cell survival, we're getting good differentiation into the appropriate cell type, and we're getting connectivity. You might say, wow, does that explain everything? So we also looked at all of these other effects of the cells, the ones that we'd seen in the stroke models, for example. And we see, for example, reduced glial scar, quite significant reduction in glial scar. We saw, again, local immune modulation. So you see a reduction in the M1 phenotype of cell and an increase in the M2 phenotype with the CTX cells. And here, nicely, they use the fibroblast control as well. So it's not just any cell. And we also saw neurogenesis, so an increase in the number of double Corton BRDU positive cells, and also angiogenesis, so an increase in the number of RECA1 positive cells. So again, how these cells work is, they may well be integrating, they may well be surviving, but clearly what's also important is they're doing a whole raft of regenerative things in the brain. We've also been working with colleagues in the UK, and it's Clive Ballard and Martin Brostock at KCL, at the various Alzheimer's models. And here we see actually rather poor cell survival. So there's not that many cells around post-mortem, albeit that you have to leave them in there for an awful long period of time. But we do see other reparative things that you might expect are important in an Alzheimer's model. And that means, for example, an increase in VEGF and BDNF, and an increase in synaptogenesis in the brain compared to the vehicle. We also see some behavioral effects, but those are not fully read out yet. And another kind of interesting aspect that's close to my heart is working with James Phillips at UCL, looking at an artificial nerve model. So this is producing an artificial nerve that could be used in peripheral nerve injury. And we published this recently to show that you can actually get an increase in muscle mass, you can get an increase in electrophysiology compared to an autograft with this artificial peripheral nerve. And we're doing a further follow-up study, a sort of proof of concept study, as a company. So there's now a company called CALIN, which is funded by UCL Ventures, the Rainbow Seed Fund, and Innovate UK now. So let me get on to stroke, which is the business today. So stroke is a huge area. This is all very familiar to you. There are no treatment options available, particularly for chronic stroke patients that have a disability. And that's what we're trying to target with these cells. This is a kind of well-used slide which shows all the opportunities for cell therapy treatment. So if you go in, so the only approved treatment is thrombolysis or thrombectomy. That you have to do that within a few hours. Within a couple of days, maybe max, you can look at affecting the immune system so that you can, improve or prevent the long-term loss of brain tissue. But then beyond that, you're really stuck with the only possible therapy is brain repair. So here you can do everything, or potentially do everything peripherally. But here you really have to look at the brain rather than the vascular system for treatment. And that means you have to inject cells locally, almost certainly. So we published our first paper a couple of years ago on the phase 1 trial. And this was a phase 1 dose escalation safety study with 24 months follow-up. There were 11 disabled stable stroke patients, 6 months to 5 years after stroke, with an average of about 2 years after stroke. It was a stereotaxic implantation. We have our own approved injection device that we developed, and it's approved as a class Mark 3. And we use doses of 2, 5, 10, and 20 million cells. Key thing here was safety. There were no cell-related or immunological adverse events. The patients weren't treated with any immunosuppressive drugs because that would be clinically high risk. And there was no evidence of any antibody formation to the cells. And we saw significant improvement in the NIH stroke scale. So they did get better as a group. And 3 of the patients improved in the modified Rankin score. And I'll go into that in more detail because it's now become our main measure of improvement. In Pisces 2, which is another UK trial, again, this was an open label trial, not yet published. And it was really based on trying to get a little bit more information. So going earlier into patients, rather than going, what we were about 2 years post-stroke, we wanted to look at patients in the rehab phase. So 2 to 12 months is where we ended up. And we also looked at patients with a specific motor deficit, an inability to move the arm or the hand. So this was really looking for any potential for functional improvements of a specific nature, as well as the more global improvements you might hope for. So there were, so these are the inclusion criteria, and they had to show upper limb dysfunction because there's a particular test, it's called the Action Research Arm Test, specific for the upper limb, but there's a particular sub-test where the patient has to pick up a block and place it in on an elevated shelf within a set period of time. And the patients were recruited who couldn't do that task. And we looked at a number of measures, some of them more general measures like modified Rankin and Bartel, but we also looked at motor tests such as Arrett and Fugel-Meyer. 23 patients were treated across 8 centers, and all of the 8 centers did their own neurosurgery, so it's it can be done anywhere. It's not a specialist treatment. And there's the sort of demographics of the patients. And what we saw was responders. So we defined responders. We looked at responders, and they were defined in this way. And at 3 months, we ended up getting what we wanted to get, which was 3 out of 20 at 12 months. We didn't get it at 3 months, but we did see it at 6 and 12 months. Interestingly, what we also got, which we didn't expect, was a considerable number of improvers in modified Rankin and Bartel, much more than you would have expected from this particular population. Now, without a control group, you can't say anything definitive about that, but it was encouraging. And it's really altered our perception as to what we do next. The other thing I think that's important as well is, I mentioned this business about doing use of the upper limb. We wanted to originally select patients that had some twitching in the upper limb. In other words, they could make a shivering response without necessarily being able to move their arm. But a lot of the sites were having difficulty recruiting those patients. So we ended up choosing patients that couldn't do that. So the NIHSS motor arm test gives you a measure of that effectively. So 4 means that you get no movement in the upper limb. So that's what we wanted to avoid originally, but we had to accept that to a degree. And so we were looking for patients with 2 or 3. So they had some twitching, but not any ability to do anything functional with that arm. And we found that produced that had quite a dramatic effect. So if you had some movement, you got much better performance on the ARAT test, and you also got, surprisingly, you also got improvements on the modified Rankin, which is a measure of overall disability. So if there's some ability to respond on the upper limb for some reason or other, that means the cells will work better for you in terms of their ability to improve your overall level of disability. So that gave us a bit of food for thought, but nevertheless, I think it's certainly, taught us a lot about how we should do a future study. So again, we were surprised by some of the data we got. Again, with the limitation that, of course, this was an uncontrolled trial. The intracerebral injection was well tolerated, able to be done at a number of different sites. And the only adverse events were surgical procedure or just normal complications of stroke. Okay, and we decided also to focus on the patient subgroups with the strongest response in this trial. So Pisces 3 comes along with its own logo. We have an IND approval to begin this trial very soon. It's close to starting, I would say, in the next 2 or 3 months. It is a randomized, placebo-controlled phase 2b study. Patients had to have an ischemic stroke 6 to 12 months previously. Modified Rankin score of 3 or 4. Okay, that's the sort of middle level, mild to moderate disability, not the worst. And I'll show you a little bit more of this later. And also have that residual upper limb movement, so have a 2 or 3 on the NIHSS motor arm test. And the primary endpoint will be a response by the modified Rankin scale 6 months post-treatment. So shifting one level at least in this disability scale. And I'll tell you how important that is in terms of both clinical and commercial value. And a number of key secondary endpoints as well, which I won't go into. And so we are recruiting now. US sites use a hub and spoke. Now that means we have, because it's a randomized control trial, how are you going to control for the fact that you're doing a surgical procedure? Well, we're going to do a surgical procedure on the placebo patients, but they're not going to get a full burr hole. They're just going to get an indentation in the skull, and they certainly won't get anything injected. But, they're going to have a half an hour operation, whereas the patients that get the cells will have a 2-hour operation. So you can't really blind the surgical team. So we're using separate surgical sites from recruitment and testing sites. And that's for all these reasons. We need to improve masking of the endpoint assessment and also provide a system efficient system for getting patients treated. It'll be 110 patients, 1-to-1 randomization, 20 million CTX cell dose as used before. We're assuming a 12.5% placebo rate of response. And that's based on data around both cell treatments in stroke patients as well as other forms of treatment for stroke patients. So there is another company doing something similar, a company called Sanbio in the US. And they've recently done a phase 2 trial with their cell therapy. And they got no effect on modified Rankin, no effect at all. That wasn't their endpoint, but they were measuring it. So that's encouraging because it doesn't mean that modified Rankin is something that necessarily responds to a placebo. But nevertheless, we have to make an assumption. So we're looking to assume a 35% response rate in our treatment group versus 12.5% placebo, which would be a significant improvement. And we power it accordingly. And of course, so important is training, certification of investigators, centralized review of the data, particularly the modified Rankin patients. So they'll be videotaped during the assessment, and that will be all of the assessment effectively will be done by a central group. Okay. So modified Rankin scale. So what is it? It's a very simple measure. It actually starts off at 6, which is dead, and goes down to 0, which is normal. And in between, you see all the variations. And of course, these are very classic things. So if you're bedridden, completely dependent on others, you're very high. One step down is you're in a wheelchair. Another step down is you've got a stick and maybe somebody to help you out when you need to go to the toilet. 2 is, you've got a stick, but you can do everything by yourself, get in the bath. And 1 is effectively normal. So what we're looking to do is recruit people at 4 and 3 and have them to drop by 1 point if we can. Now this is, it's a simple clinical observational test, and it's done in a relatively formalized way. So it's not just, somebody making a judgment without having to go through a thinking process. And what does this mean commercially? And this is interesting. This was some data published last year by a Swedish group looking at the cost of care for patients at these different levels of the modified Rankin. And so, if you show a shift from 4 to 3, you've got a massive change from $90,000 to $50,000 per year. $40,000 saving per year in treatment. Slightly smaller, but nevertheless significant for MRS3 and MRS2. So the commercial case is there, and the likelihood of getting registration if you get a successful outcome is also high. Great, so I've had a big team over the years. This is a relatively small part of that big team, and these are all the people that I work with directly, particularly in the isolation discovery and initial clinical work with CTX Solar. So thanks very much.
Thank you. Are there any questions?
Hi, thank you. That was a very interesting talk. I had a question about your dose. In your early study, you had done 4 doses, I think. Did you pick the highest subsequently?
Yes.
20 million?
Yeah.
Is that the highest you've looked at?
No, well, yes, it is the highest we've ever looked at. And the reason for choosing that dose is not just because we wanted to give the highest safe dose. We had to think of the cost of manufacture and what it would cost us to double that dose, for example, in terms of manufacturing, therefore ultimately the cost of goods of the product. So there's a bit of, we did a lot of pharmacoeconomics around, in the past, around, how much is this worth, for what level of improvement, in what patient subgroups, etc. And obviously in stroke, as you might imagine, it's much more cost effective to treat younger stroke patients than older ones, because obviously if you can get patients back to work. I mean, one of our phase 2 patients was a builder who had a stroke in his 40s. And, last I heard, he was back working, from being effectively wheelchair bound, he was back working. So, what do you make of it? It could have happened by chance, but, interesting.
You mentioned it's a 2-hour procedure, the delivery of the cells.
Yeah.
So therefore, it's a major medical intervention.
Yes.
Requiring a major hospital environment.
Yeah.
Is there any prospect of?
Actually, in the US, they do it in using local anesthesia, maybe with the patients on a, dissociative. But yeah, so it's not, I don't think it's that major. It just takes a time. It's really the rate at which you can inject cells into the brain. Yeah.
Why it gets on.
Yeah. Yeah.
The flow.
Yeah, it's, you can't, obviously you want to avoid shear forces in the injection process if you can.
Can I just ask you, the preclinical models with exosomes as opposed to stem cells to get your target results?
In stroke?
Yes.
We are doing a preclinical study in stroke, yeah. But we have no results from that yet. It's underway.